Khatib series in Human Anatomy and Physiology. Chapter 2, Cell Structure and Function. Episode 8, Cell Transport. Part 3. Bulk Transport, Endocytosis and Exocytosis. If you enjoyed the video, don't forget to hit the like button and share it, to help us reach every medical and health professional student around the world. And of course, make sure to subscribe to the channel for more valuable content. Your comments are highly appreciated. In our previous videos, we explored the different mechanisms of passive transport across the cell membrane. Today, we're shifting focus to another vital process that is the bulk transport. Bulk transport is essential when cells need to move large particles, whole microorganisms, or large volumes of fluid in or out of the cell. These substances are too big to pass through traditional transport methods like diffusion or facilitated diffusion. This transport mechanism plays a critical role in many cellular activities. It allows cells to absorb large nutrients, eliminate harmful materials, release hormones and enzymes, and even defend the body by removing pathogens. In addition, like active transport, bulk transport is a form of active transport, meaning it requires energy, usually in the form of ATP. Bulk transport has two main types. These are endocytosis, which is the uptake of materials into the cell, and exocytosis, which is the release of materials out of the cell. Let's begin with endocytosis. As we said, endocytosis involves the formation of vesicles to bring substances into the cell. The process starts when the cell detects a substance near its surface. Then, the plasma membrane folds inward, forming a pocket around the substance. As the pocket deepens, it eventually pinches off, forming a vesicle that carries the material inside the cell for further use or processing. In all eukaryotes, endocytosis comes in three main forms. These include phagocytosis, pinocytosis, and receptor-mediated endocytosis. Let's start with phagocytosis, which is often called cell eating. In phagocytosis, the cell engulfs large solid particles, like bacteria or cellular debris. This process is especially important in immune defense. Indeed, specialized immune cells like macrophages and dendritic cells perform phagocytosis to capture and destroy pathogens. As explained in earlier episodes, the process of phagocytosis begins when the cell detects a pathogen. The membrane then wraps around it and forms a vesicle called a phagosome. The phagosome then fuses with a lysosome, forming a phagolysosome, where the pathogen is digested by digested enzymes. The cell utilizes some substances. Any leftover waste is then expelled from the cell by exocytosis. Imagine this animation and see how a white blood cell engulfs a bacterial cell during phagocytosis. On the other hand, pinocytosis, or cell drinking, is the process where the cell takes in droplets of extracellular fluid along with its dissolved nutrients. These are dissolved solutes such as sugars, ions, and proteins. This allows the body to efficiently absorb nutrients that are dissolved in the intestinal fluid, even when they are present in very low concentrations. Like other types of endocytosis, the process involves detection, then pocket formation, followed by vesicle formation. In the initial stage of pinocytosis, the cell detects the presence of extracellular fluid around its membrane. This process does not require specific receptors or signaling molecules. Instead, the cell membrane begins to invaginate, forming a small pocket that captures the surrounding fluid and any dissolved substances in it. As this vesicle is pinched of, it forms a vesicle called the pinosome, containing eth fluid and its dissolved substances. This vesicle fuses with the lysosome to form pinolysosome for further degradation. Like other types of endocytosis, the cell utilizes beneficial materials and any indigestible materials are excreted by exocytosis. This electron microscopic image demonstrates the formation of pinosome vesicles during the transport of materials in the cell. A more selective type of endocytosis is called receptor-mediated endocytosis. In this process, cells use specific receptors on their surface to capture and internalize molecules or ligands. One example of molecules that use this method of cellular transport is low-density lipoprotein cholesterol, which is transported to cells via specific receptors called LDL receptors. Another example is the transport of iron into the cells via the transferrin receptors. In general, membrane receptors are found in regions of the plasma membrane called pits, which is very rich with protein clathrin. This protein is important for the formation and pinching of the vesicle, as well as in transporting the vesicle between different intracellular compartments. Once the ligand binds to its receptor, the pit regions are internalized, and clathrin-coated vesicles are formed. The clathrin-coated vesicle fuses with other endosomes. The receptor is isolated and the vesicle is uncoated, form clathrin by special intracellular proteins such as heat shock protein 70 and auxilin. The vesicle then fuses with the lysosome for further degradation as in the remaining types of endocytosis. This electron microscope image shows clathrin-mediated endocytosis of an adenovirus into the cells. Notice the internalization of virus through clathrin-coated pits into clathrin-coated vesicles. This table summarizes the different types of endocytosis and the variations in their mechanism in transport of materials with some examples. Opposing endocytosis is exocytosis. Exocytosis is a vital process for the cell where wastes and many large biological molecules are needed to be moved out the cell. Examples of secretory materials include hormone secretion into the bloodstream, the secretion of digestive enzymes into the lumen of organs, the release of the neurotransmitters form neurons, and getting rid of wastes. In this process, vesicles formed inside the cell carry substances to the cell membrane. Once at the edge, the vesicle fuses with the membrane and releases its contents into the space outside the cell.